Cancer IV drips and the glutathione vitamin C connection. High-dose intravenous vitamin C can act as a pro-oxidant in killing cancer cells, where it has to be separated from glutathione. Otherwise, vitamin C needs to be kept in balance with glutathione. So what does this mean? Well, make sure you watch this now because this is only available for free for 48 hours and then it is reserved for MasterPass members. Let's get down to business. Is high-dose vitamin C good for you? Well, high-dose intravenous vitamin C can selectively kill cancer cells in live patients and can save sepsis patients from dying, but it acts as a pro-oxidant in cancer and an antioxidant in sepsis. So what does that mean for the rest of us? Oral doses of 2,000 milligrams of vitamin C per day raise oxalate levels in most people, and as little as 400 milligrams, possibly lower, raises oxalate in some people. So what does that mean? Is it about the dose or not? Well, it's about the dose and the person and the context and all the stuff underneath the hood. This seems to be, meaning oxalate, seems to be the most sensitive indicator of a delicate imbalance with glutathione and other factors needed to recycle the vitamin C. Such a balance actually needs to be avoided when killing cancer, yet it is critical to maintaining health in every other context. Individuals under high stress appear to need at least 250 milligrams beyond diet to minimize the incidence of colds, and anecdotally, some people seem to benefit from much higher doses. But how do we take advantage of such benefits without upsetting the delicate balance with glutathione and the propensity to generate oxalate? Well, that, my friends, is the topic of this episode. But this episode, past this point, is only available for free for 48 hours. It is, after that, reserved for MasterPass members. So check the link in the description to find the written article full with references, as well as the evergreen video and podcast. And with that said, let's get down to business. Vitamin C recycling and degradation. We need some background here in order to understand this topic. Apart from being an enzymatic cofactor for a variety of different iron and copper dependent enzymes, vitamin C acts as an antioxidant where it fulfills a niche within the broader antioxidant system. Vitamin C, ascorbic acid, dare it, I said it, vitamin C is ascorbic acid, functions as an antioxidant because it has 2 electrons that it can give up. These electrons help quench the fire of oxidative stress. They can be used to recycle vitamin E when it is oxidized or to directly scavenge oxidants, putting out the little flames before a fire starts. When ascorbic acid gives up 1 electron, it becomes a free radical known as the ascorbyl radical. Electrons have a voracious appetite to exist in pairs, and a free radical is something with an unpaired electron. All free radicals are highly reactive because they want to pair up their unpaired electron. However, the ascorbyl radical is relatively stable compared to the radicals quenched during its formation, and it is easily recyclable using a number of enzymes. The formation of the ascorbyl radical therefore safely transfers the burden of satisfying the voracious pair bonding appetite of electrons away from the delicate structures of the cell that could be damaged by this appetite and onto the enzymatic system of antioxidant defense. Enzymatic recycling of the ascorbyl radical uses electrons carried by NADPH or NADH. NADPH and NADH are both forms of niacin, also known as vitamin B3. NADH is used by the riboflavin-dependent enzyme cytochrome B5 reductase, which animal experiments suggest is also dependent on calcium and magnesium. NADPH is used by the selenium and riboflavin-dependent enzyme thioredoxin reductase. NADPH gets its electrons from glucose in the pentose phosphate pathway, and NADH gets them from any type of food energy. Thus, the overall rate of energy metabolism is critical, and things that hurt it, like diabetes, thyroid disorders, adrenal disorders, deficiencies of other nutrients involved in energy metabolism, or the 1,451 inborn errors of metabolism, the prevalence of carrier status of which is 100%, collectively, so everyone has these key bottlenecks in their metabolism. All these will hurt vitamin C recycling. So remember, if you don't know that the prevalence of rare things is 100%, go see my Super Unlock video. All right. While recycling by NADH or NADPH is the most common path for the ascorbyl radical, it can also give up a second electron to become dehydroascorbate, or DHAA. In fact, vitamin C can recycle itself in a process known as disproportionation, where two ascorbyl radicals partner up. One takes an electron from the other to become the fully recycled ascorbic acid, and the other loses that electron to become the fully oxidized DHAA. DHAA can then be recycled by glutathione or NADPH. Glutathione can recycle it without an enzyme, but this doesn't happen fast enough to prevent it from irreversibly degrading. Somewhere between 67 to 75% of the recycling depends on glutathione and uses enzymes such as glutaredoxin, protein disulfide isomerase, and a specific form of glutathione S-transferase. Glutathione itself is made in a magnesium and energy-dependent manner and gets recycled by NADPH and thus ultimately from glucose, again placing emphasis on the importance of energy metabolism and healthy glucose levels. Some 25 to 33% of DHAA recycling depends on NADPH directly and uses enzymes such as the selenium and riboflavin-dependent enzyme thioredoxin reductase or 3-alpha hydroxysteroid dehydrogenase. Whenever DHAA escapes through this recycling network, will react with water to irreversibly degrade into 2,3-diketogulanate. Some of this is excreted directly into the urine. However, some of it will always degrade further, and in the presence of hydrogen peroxide, which there is always some of in the cell, 2,3-diketogulanate further degrades into L-threonate. In the absence of hydrogen peroxide, it generates L-erythrulose, and regardless of which path it takes, it always generates oxalate. Thus, the generation of oxalate from vitamin C represents the failure of a two-step defense system. Because the ascorbyl radical failed to be recycled with the help of riboflavin, niacin, selenium, calcium, magnesium, glucose, and energy, the recycling burden fell to the glutathione-dominant second step. This second step also failed, and the DHAA was irreversibly hydrolyzed, meaning broken down by water, to form oxalate. Now, if you've also seen my recent writings or videos or podcasts on oxalate detoxification, it is my hypothesis that oxalate can be detoxified in a two-step process. So you have a failure of two steps to recycle vitamin C that generates oxalate. Now you have a failure of two steps to get rid of the oxalate by converting it to carbon dioxide. My hypothesis is that the biotin and manganese-dependent enzyme pyruvate carboxylase converts oxalate to formate, and that formate then has multiple fates, which include dehydrogenases that can get rid of it as carbon dioxide and also includes utilization in other pathways, especially the methylation pathway. And so a failure of all of those potential fates of formate, or from the first step of converting oxalate to formate, would make the oxalate build up more when it is generated by the failure of the two steps of the vitamin C recycling pathway. So we can add that in here as well. All right, now, energy pathways implicate even more nutrients. The importance of food energy to generate NADH implicates all of the B vitamins, all of the electrolytes, salt, potassium, calcium, magnesium, and bicarbonate, iron, copper, sulfur, and magnesium. The importance of the pentose phosphate pathway to generate NADPH implicates glucose, thiamine, vitamin B1, calcium, and magnesium. It also implicates glucose 6-phosphate dehydrogenase deficiency, which impacts about 8% of the world's population and compromises the supply of NADPH. Now, all over the place, you see it acknowledged that glucose 6-phosphate dehydrogenase deficiency compromises the ability to tolerate vitamin C. But what about the 1,450 other inborn errors of metabolism? And they might not be common in their diagnosable defect manner, but if carrier status is a 50% bottleneck on the metabolic pathway, and if everyone's carrying a couple of them and they can interact to produce the full-blown effects of a disorder that would never be diagnosed because of the diagnostic criteria and its failure to capture synergistic heterozygosity, and if also the 50% bottlenecks, even when they don't interact to produce a full-blown disorder, could produce some kind of intolerance to 2,000 milligrams of vitamin C as a result of limiting the recycling, then you simply have a failure of being biochemically optimized, and it's not a diagnosable anything, nor is it that severity, but you might not feel great, or you might worsen your health, or you might cause gray hair or whatever by supplementing with too much vitamin C. And these are the many rare idiosyncratic things that collectively impact everyone. The possible exception of the 120-year-old woman living off bacon grease and cigarettes. Okay. Glutathione is synthesized using food energy and recycled by NADPH, implicating all of the above. Since glucose is so critical to the pentose phosphate pathway, hypoglycemia is a particular risk. When the glucose is derived from glycogen breakdown, vitamin B6 becomes especially important. And when it's made anew from protein in gluconeogenesis, biotin, potassium, magnesium, and manganese become especially important. Now, does vitamin C supplementation raise oxalate production? Studies on vitamin C supplementation and oxalate are few and far between, but generally supportive of high-dose vitamin C raising urinary oxalate levels. In one study that was elegantly controlled for using different doses, all taken as 2 divided doses across the day, 1,000 milligrams per day consistently raised urinary oxalate, whereas 400 milligrams per day raised it markedly in some individuals, but not enough to carry up the group average. Doses between 30 milligrams and 200 milligrams did not seem to cause any major changes in oxalate for anyone. As elegantly as this study was when it came to the dosing, they measured urinary oxalate right after the subjects came off of a scurvy level depletion diet rather than at the end of the 180-day study, which strikes me as a serious methodological error. The subjects were likely suffering from elevated oxidative stress, making them more sensitive to the oxalate rise, for the reasons we just talked about. Had they measured it at the end of 180 days, we would have seen the effects of long-term daily use, which would have been much more relevant. In a second study of 12 subjects who form kidney stones and 12 who don't, 6 days of 2,000 milligrams of vitamin C taken as 2 divided doses raised urinary oxalate by 33% in those who form stones and by 20% in those who don't. In a third study of 29 stone formers and 20 non-formers, 6 days of 2,000 milligrams of vitamin C taken as 2 divided doses raised urinary oxalate by 16% in stone formers. In non-formers, urinary oxalate was 7.5% higher when using vitamin C, but that was not statistically significant. Acute intravenous use of 2 grams or 10 grams of vitamin C dose-dependently raises urinary oxalate in critically ill patients. And in kidney patients on regular dialysis treatment, even 250 to 500 milligrams per week over 6 months raises blood levels of oxalate. So overall, these studies suggest that in the average person, doses of 1,000 milligrams per day or more increase oxalate, with higher confidence for 2,000 milligrams per day. Less consistently, in some people, as little as 400 milligrams per day increase oxalate. In kidney patients on dialysis, as little as 250 milligrams per week intravenously raises oxalate. So, I have not seen any studies looking at how the factors that contribute to vitamin C recycling impact this outcome, but it seems rather obvious that any deficiency or suboptimal status of riboflavin, niacin, selenium, calcium, magnesium, glucose, energy, or glutathione, and energy is a big Pandora's box, will have a major impact on who generates oxalate and how much at what dose of vitamin C. So without making any ridiculous over-extrapolation such as 1,000 milligrams of vitamin C raises urinary oxalate, it seems rather sound to conclude as follows. First, everyone should be mindful of their glutathione hygiene and their suite of vitamin C recycling nutrients when using 1,000 milligrams per day orally or more, or when using any intravenous dose. Number 2, many people should be mindful of these factors when supplementing orally with as little as 400 milligrams per day. Kind of hard to know who is who, but if you have any oxalate-related problems, be sure to put yourself in this bucket. And then finally, a few people need to bother with this when supplementing orally with 200 milligrams per day or less. There are people who need to bother about it, but it's not very many. This has remarkable correspondence to the amount of vitamin C you can get from food. 200 milligrams per day is a very vitamin C-rich diet, and it would be quite challenging to hit 400 milligrams per day. I'm not saying you can't do it. You can, but it'd be quite challenging to do that. Whereas if you're really trying to eat vitamin C from foods and you're not going crazy doing it, or you're not eating something relatively obscure, you're likely to hit 200 milligrams per day, but you're not likely to hit 400. Vitamin C as a pro-oxidant. Vitamin C can react directly with oxygen to form a peroxide. The peroxide will quickly decompose to the ascorbyl radical and superoxide. Oxygen on its own is considered so innocuous that this might as well be a case of vitamin C oxidizing itself. So this is known as the autooxidation or auto-oxidation conjoined to the more simple autooxidation of vitamin C. More technically, oxygen is so abundant in open air and within cells that its presence is a given, and autoxidation is any oxidation that merely requires the presence of oxygen without requiring any other type of intervention to make it happen. As covered in my antioxidant course, superoxide is generally the first of the oxidants to be produced and will be quickly scavenged in the mitochondria with the manganese-dependent superoxide dismutase enzyme, SOD, or outside the mitochondria with the copper and zinc-dependent SOD. This will then form hydrogen peroxide. Alternatively, vitamin C can scavenge superoxide directly, but this hardly cleans up the mess. It still leaves behind hydrogen peroxide, and it sends vitamin C into step 2 of its recycling, which, if it lags, leaves in its wake its irreversible degradation to oxalate. The hydrogen peroxide can be cleared using glutathione and the selenium-dependent enzyme glutathione peroxidase, or by using the iron-dependent enzyme catalase. These enzymes convert hydrogen peroxide to water. While copper and iron are both critical antioxidants when used as enzymatic cofactors, in free form they can worsen this damage. They can catalyze the autoxidation of vitamin C, meaning they can make it happen much faster. If they get a hold of the hydrogen peroxide before glutathione peroxidase or catalase convert it to water, moreover, they can break it down to form the infamous hydroxyl radical, a free radical far more dangerous than hydrogen peroxide or even superoxide. Glutathione prevents the autoxidation of vitamin C in purified solutions powerfully enough that at normal glutathione and vitamin C concentrations within cells, we would not expect the autoxidation of vitamin C to occur at all. The mechanism is unclear, and there may be multiple. Glutathione can chelate copper and iron, and there are usually some of these metals contaminating even the most purified water. Although I can't find any direct evidence of this, I'm tempted to speculate that glutathione neutralizes the peroxide form of vitamin C before superoxide is released. One of glutathione's major roles is to convert peroxides to hydroxyl groups. It converts hydrogen peroxide to water this way. It converts lipid peroxides to hydroxy fatty acids this way. And if it did the same thing to the peroxide form of vitamin C, it would lead to the fully regenerated ascorbic acid. While various versions of the selenium-dependent enzyme glutathione peroxidase will help this happen within living cells, glutathione neutralizes peroxides this way all on its own in vitro, just less quickly than with the enzyme. If I am correct about this, glutathione would interfere midway in the autoxidation of vitamin C and prevent the release of superoxide. And now we get to cancer. IV vitamin C as a pro-oxidant in cancer treatment. High-dose intravenous vitamin C is of interest as an adjunct to chemotherapy in some types of cancer. It is widely used by complementary medicine practitioners and supported by animal experiments, case studies, case series, but does not yet have any support from randomized controlled trials. That doesn't mean that it's BS, because evidence-based medicine doesn't look at everything, and it hasn't looked at many things. While mechanistic cellular studies are in disagreement on the exact nature by which vitamin C helps kill cancer cells, they are broadly in agreement that it is by acting as a pro-oxidant, especially by generating hydrogen peroxide. They are also broadly in agreement that the same concentrations do not kill normal cells and may actually protect normal cells from the adverse effects of chemo. This is because normal cells have better defenses against hydrogen peroxide than cancer cells do. When glutathione is combined with vitamin C, the glutathione dramatically reduces the ability of vitamin C to kill cancer cells. This is true across different types of isolated tumor cells. It is unsurprising since glutathione prevents the autoxidation of vitamin C, and to whatever extent it fails at this, it clears up the resulting hydrogen peroxide. So notice this massive difference between cancer and the usual context. In the usual context, you want to prevent oxalate generation at least by minimizing stress on the system by what you don't want is to have vitamin C out of balance with glutathione. In the context of cancer, what you exactly want is to have vitamin C out of balance with glutathione, because this is what enables it to kill the cancer cell. Remarkably, the papers on this include an animal study that shows that while glutathione injections antagonize the ability of vitamin C injections to limit tumor growth, they do not antagonize the positive effects on survival. In other words, vitamin C was very effective at limiting tumor growth and promoting survival. And when glutathione was added, the animals survived just as well, but with bigger tumors. Very interesting. The authors suggested that IV vitamin C should be given without glutathione alongside oxidant-based chemotherapy, while IV glutathione should be given in between those sessions. That way, it will allow the vitamin C to generate a large pro-oxidant effect in the cancer cells that synergizes with the oxidative chemo drugs, but helps protect normal cells. Then, the glutathione can come in later when the damage is done to the tumor and provide its own pro-survival benefits without interfering with vitamin C's pro-oxidant effect. They also included a survey of complementary medicine practitioners. 95% of them reported using IV glutathione. 89% of those who used IV glutathione also used IV vitamin C. 77% of them on the same day in the same patient, and 15% of them mixed them in the same infusion bag. Only 13% of them were using IV glutathione for cancer, so it's not clear how many were mixing the two together for cancer specifically. However, the authors recommended greater awareness on the ability of vitamin C and glutathione to antagonize each other in the specific context of cancer. Let's shift gears now. IV vitamin C is an antioxidant in kidney disease. In kidney patients on chronic hemodialysis, antioxidant support is needed because the dialysis itself causes oxidative stress. This is due to the mixing of non-biological material with blood, which activates immune cells to produce oxidants. A randomized controlled trial found coating the dialysis membrane with vitamin E and infusing 1 gram of vitamin C over 4 hours abolishes the generation of hydrogen peroxide and the oxidative damage that results from it. As reviewed in the literature, numerous studies have looked at this and are in broad agreement that IV vitamin C either has no effect on oxidative stress or has antioxidant effect in hemodialysis patients. It doesn't have a pro-oxidant effect. However, most of these studies are using relatively low doses, such as 250 or 500 milligrams, infused intermittently in association with the dialysis treatment, often being once a week. 250 or 500 milligrams of vitamin C infused once a week is not a high dose. We're going to come back and revisit these doses after we discuss sepsis. So now let's talk about vitamin C and the oxidative burst. One other case where there's randomized controlled trial support for IV vitamin C is in sepsis, which is an extreme inflammatory response to infection. It is possible that vitamin C's promotion of immunity to infection underlies its benefit in sepsis. As covered in several papers, the role of vitamin C in immunity is largely, paradoxically, to promote the production of oxidants by acting as an antioxidant. The oxidative burst is used by a variety of immune cells, especially neutrophils. It helps kill pathogens. Its end goal is to produce superoxide, nitric oxide, peroxynitrite, hydrogen peroxide, and hypochlorous acid, which is also known as bleach. All of these are antimicrobial. It achieves this using the help of niacin, glutathione, vitamin C, all the vitamin C recycling nutrients that we discussed earlier, the amino acid arginine, oxygen, zinc, copper, iron, and chloride from salt. The initial products are nitric oxide and superoxide. The superoxide generates hydrogen peroxide. The superoxide and nitric oxide combine to form peroxynitrite, and hydrogen peroxide combines with chloride from salt to generate bleach. The nitric oxide, which also gives birth to the peroxynitrite, is made by nitric oxide synthase, whose cofactor BH4, or tetrahydrobiopterin, is extremely sensitive to oxidation. The specific role of vitamin C in this arrangement is to serve as an antioxidant that protects BH4, defending it against extremely oxidative environments in which it needs to function. In other words, vitamin C is enabling the BH4 to be preserved, acting as an antioxidant in order to enable the BH4 to contribute to the massive amounts of oxidants that are swimming all around and which could come back to bite the BH4 in the butt. Vitamin C will need to be recycled, and that falls on the suite of cofactors and enzymes involved in the two-step recycling process that we discussed earlier. With the deliberate proliferation of oxidants, immune cells are especially in need of antioxidant protection. You don't go to war without wearing armor. So vitamin C functions along with the entire antioxidant defense to protect immune cells against their own weapons of mass destruction. This is important to keep in mind because even though vitamin C is enabling the production of oxidants, we don't actually want it autoxidizing and forming superoxide and hydrogen peroxide, both in close proximity to the BH4 cofactor of nitric oxide synthase and near the delicate structures of the immune cells. We want it acting as an antioxidant, which means that we don't want high-dose IV vitamin C if it's going to cause the same effect as it causes in cancer, but it doesn't. So let's talk about IV vitamin C in critical care patients. One very special consideration for patients with sepsis and other critically ill patients who receive IV vitamin C is that most of them are deficient in it. One important study on this topic was the Citrus Alley randomized controlled trial. ICU patients with sepsis were randomized to receive either IV vitamin C 50 milligrams per kilogram body weight per every 6 hours for 96 hours, or placebo. For a 70 kilogram standard reference man, this would be 3,500 milligrams every 6 hours for a series of 16 doses. Roughly 75% of the subjects had vitamin C below the normal range at the start of the study. In the placebo patients, it stayed there. In the vitamin C patients, most of them entered or exceeded the normal range for the duration of the treatment, and more than 50% of them stayed in the normal range even 3 days after the treatment ended. The study was designed to look at inflammatory markers and organ failure scores as its primary endpoints and found no effect. It had 46 secondary outcomes, and in 43 of them, there was no difference. Nevertheless, in the vitamin C group, mortality was 34% lower. Release from ICU within a week was twice as common. Hospital-free days were 45% more common, and ventilator-free days were 24% more common. These were only statistically significant if they treated each comparison on its own rather than accounting for making almost 50 of them. As such, we need to regard this as a suggestive benefit, not a statistically robust one. Despite the suggestive nature of the statistics, all the differences are in the direction of a benefit for vitamin C. And frankly, they would have been well justified in pre-specifying 28-day mortality as their primary endpoint. I don't know why they didn't. Relegating this endpoint to a series of nearly 50 secondary analyses seems like a case of the soft bigotry of low expectations. So let's take this as an important demonstration of a potentially huge benefit for vitamin C in sepsis patients. But now let's compare the blood levels reached in these different uses. Comparing the plasma levels for different uses of IV vitamin C. So, what are we actually achieving with various doses of IV vitamin C? The concentration needed to kill 50% of cancer cells in vitro ranges from 500 to 3,000 micromoles per liter, depending on the type of cancer. Presumably, this is in the range of plasma values that would prove successful in trials and is also where a physiologically meaningful pro-oxidant effect begins to take place. A 70 kilogram standard reference man in the Citrus Alley study would be getting 3,500 milligrams of vitamin C infused every 6 hours for a series of 16 infusions, leading to an average peak plasma value of only 169 micromoles per liter. In healthy subjects, a single IV dose of 3 grams produces an average peak plasma level of 1,760 micromoles per liter, while a single IV dose of 5 grams yields 5,580 micromoles per liter. In kidney patients, usually much lower doses of IV vitamin C yield plasma levels on the order of 23 to 113 micromoles per liter. That is, in sepsis patients, where there's a strong suggestive benefit, the average levels reached with repeatedly dosing 3.5 gram infusions never leave the normal range and never get anywhere near the level needed to kill off cancer cells. This is because sepsis patients are deficient in vitamin C and their bodies are voraciously consuming it. In dialysis patients, the doses that are used are small and intermittent, and the starting plasma values are mediocre or deficient, and what results is a less deficient plasma level, or a plasma level that is in a better place within the normal range. In stark contrast to both of these situations, in healthy subjects, a single dose of 3.5 grams shoots plasma levels well into the range that are needed to kill cancer cells, which are pro-oxidant. In short, IV vitamin C is likely to be beneficial in cancer because it achieves a pro-oxidant effect by bringing vitamin C way outside the normal range. It exerts a pro-oxidant effect in people who need a pro-oxidant effect. IV vitamin C is likely to be beneficial in sepsis because it brings otherwise deficient vitamin C levels into the normal range. IV vitamin C appears to be largely beneficial in dialysis patients because it brings deficient or mediocre levels a little higher in close proximity to the dialysis session, which is an acute event that provokes oxidative stress. These levels never exceed the normal range. Now, is IV vitamin C likely to be beneficial in other people who do not have any illness that is making them deficient in vitamin C? Probably not at the doses used in sepsis or cancer patients. This would likely have a pro-oxidant effect in someone who doesn't need it. If this does indeed have a benefit, it's probably a hormetic benefit rather than a nutritional benefit, meaning the vitamin C would cause acute oxidative stress, and when it's gone, the person would develop better defenses against oxidative stress in response. However, there's nothing obvious about this, and it is something that deserves to be treated carefully and studied closely. Are there demonstrated pro-oxidant effects of vitamin C in humans? There aren't any clear, unambiguous demonstrations of high-dose vitamin C causing oxidative stress outside reports of hemolysis in infants with glucose 6-phosphate dehydrogenase deficiency in premature infants, and a report of a fatal cardiomyopathy in a man with hemochromatosis. However, this is not something that is all that well studied. One paper showed that in both sepsis patients and healthy controls, acute infusion of 1 gram of vitamin C increases ascorbyl radicals in plasma. I don't find this a convincing demonstration of oxidative stress. Ascorbyl radicals may reflect the autoxidation of vitamin C and thus the generation of superoxide and hydrogen peroxide, but they're also formed when vitamin C acts as an antioxidant or as an enzymatic cofactor. In fact, healthy controls had greater concentrations of ascorbyl radicals in their plasma than sepsis patients did, and plasma vitamin C levels explained 73% of the variation in ascorbyl radical levels. This to me just means vitamin C was doing its job. One small study of 6 healthy males showed that 6 days of daily IV vitamin C using either 750 milligrams or 7,500 milligrams per day did not alter vitamin E levels, markers of lipid peroxidation, or markers of DNA damage. And this is not really a good set of measurements. In my antioxidant course, I begin by differentiating between oxidative stress and oxidative damage. Lipid peroxidation and DNA damage are examples of oxidative damage. Examples of oxidative stress are compensations within normal systems that have adverse consequences. The prototypical example of this would be impaired glucose tolerance and elevated free fatty acids. Cells experiencing oxidative stress compensate by rejecting fuel and decreasing their energy expenditure, since burning fuel generates oxidants. This results in fuel molecules such as glucose and fatty acids accumulating in the blood. If the lipids aren't being damaged by the hydrogen peroxide generated from the autoxidation of the vitamin C, then vitamin E isn't going to be depleted. The only well-established role of vitamin E is to break lipid peroxidation chain reactions in cell membranes. More to the point, vitamin C's place in the antioxidant system is to recycle vitamin E. Even if vitamin C were increasing the work vitamin E needed to perform, it would be there to recycle it. It would be quite surprising if vitamin C depleted it. By contrast, vitamin C is recycled by glutathione. This means vitamin C will tend to oxidize glutathione. If it does this faster than the recycling of glutathione can keep up, we would expect the glutathione pool to be oxidized. That is, we would expect to find a shift from reduced glutathione, or GSH, in plasma to oxidized glutathione, or GSSG, in plasma. So the best measurements to test for oxidative stress markers in response to high-dose IV vitamin C, in my opinion, would be oxidation of the glutathione pool and impaired glucose tolerance, not vitamin E, not lipid peroxidation, and not DNA damage. But let's put it this way. If these doses of vitamin C cannot generate any oxidative stress, there's no hope for the role of IV vitamin C in cancer. All right? With that, let's now come back full circle to the issue of nitric oxide toxicity that I've raised in past writings. This is a concern for stuff that we shouldn't talk about, YouTube and all instances of inflammation. So can vitamin C worsen these things? So first, we take a deep dive into the way that vitamin C, glutathione, and nitric oxide interact. When glutathione encounters nitric oxide, it reversibly binds it to form a reservoir that can be released as needed. If vitamin C is added to the mix, vitamin C will release the nitric oxide more quickly. When vitamin C and nitric oxide were mixed together all on their own, vitamin C made the nitric oxide disappear. Specifically, when 4 micromoles per liter of nitric oxide were mixed together with 1,000 micromoles per liter of vitamin C, virtually all of the nitric oxide disappeared within 48 seconds. When glutathione was mixed in an equal concentration as vitamin C, it provided 28% protection against the loss of nitric oxide. When SOD was mixed in, superoxide dismutase, it provided 55% protection. An iron-specific chelator provided 36% protection, while a copper-specific chelator did not provide any protection at all, suggesting that the solutions were contaminated with some iron but not any copper. These results are best explained as follows. When vitamin C autoxidizes, it generates superoxide. Iron or copper, if present, will hasten this. Glutathione will prevent it. Once it happens, SOD will clear the superoxide. Whatever superoxide makes it past these defenses will react with the nitric oxide to form much more dangerous peroxynitrite. Under oxidative conditions, nitric oxide can form the much more dangerous nitrogen dioxide. Both peroxynitrite and nitrogen dioxide cause serious damage to cellular proteins. However, in a living cell with a well-balanced antioxidant defense system, vitamin C is likely to protect against nitric oxide toxicity by playing its role to minimize the accumulation of oxidants and to help keep nitric oxide intact. Vitamin C can even scavenge the oxidized forms of nitric oxide and protect against them. So, can vitamin C worsen nitric oxide toxicity? I have in the past cited a study showing that glutathione and its precursor N-acetylcysteine protect against nitric oxide toxicity in a dopamine-responsive brain cell, while ascorbic acid worsens it. The worsening effect seems to kick in robustly at 400 micromoles per liter, which incidentally is just under the minimal concentration needed to start seriously killing cancer cells. And there are some hints that it may begin around 200 micromoles per liter, which is significantly above the normal range of plasma levels. I've suggested in the past that it did this by depleting glutathione levels. I would revise this somewhat now and expand it. Vitamin C did not affect the measured glutathione levels, but we'd expect vitamin C to liberate nitric oxide from the portion bound to glutathione. This should increase the measured glutathione levels, since it didn't in that study, this does suggest that the vitamin C depleted the glutathione to some extent. The liberation of nitric oxide from glutathione would itself increase the toxic load of that substance. I looked for other papers on how vitamin C impacts nitric oxide toxicity. At first glance, they're all over the map, but there does seem to be some common threads. So first, when cigarette smoke, which is abundant in nitric oxide, is incubated with the amino acid tyrosine, which is very vulnerable to nitric oxide-induced damage, vitamin C was more protective than glutathione. However, this could have been due to neutralization of the many other oxidants in cigarette smoke that would have rendered the nitric oxide more harmful. Number 2, using isolated proteins, vitamin C and glutathione were similarly effective at protecting against peroxynitrite toxicity. The vitamin C was effective up to 1,000 micromoles per liter. However, this study used peroxynitrite rather than nitric oxide. Thus, the adverse effects of the oxidation of vitamin C were largely irrelevant, and its ability to scavenge peroxynitrite likely won the day. Number 3, using isolated DNA, dopamine, which itself easily autooxidizes like vitamin C, and nitric oxide caused strand breaks. Massive amounts of vitamin C, 10,000 micromoles per liter, proved protective. However, in this system of isolated DNA, there's no glutathione to deplete, and it is rather divorced from the context of a living cell. The autooxidation of dopamine might have provided all the superoxide needed to form peroxynitrite, and the scavenging effect of vitamin C might have been its only relevant part to play. Next up, using isolated proteins and physiological concentrations of copper ions, nitric oxide caused more damage to proteins in the presence of vitamin C, with the effect starting around 500 micromoles per liter. This is consistent with copper catalyzing the autooxidation of vitamin C, which is present at concentrations known to be pro-oxidative. In the insulin-producing pancreatic cells of rats, vitamin E protected against DNA strand breaks and cells dying by splitting apart, which is called cell lysis, and vitamin C had only a tiny benefit and glutathione did nothing. Clearly, the damage in this model was driven by lipid peroxidation and vitamin E was most helpful, so it rendered the interactions with vitamin C and glutathione less relevant. In cells that line the blood vessels of rats, vitamin C protected against nitric oxide toxicity, but the concentration used was 100 micromoles per liter, which is within the normal range of human plasma. In dopamine-producing neurons taken from rats exposed to toxic doses of nitric oxide, 400 micromoles per liter of vitamin C had slight benefit, and it was not statistically significant. However, the nitric oxide in this study was provided using S-nitrosocysteine. This would leave behind cysteine that could be used to make glutathione. The glutathione may have eliminated any negative effect of the vitamin C. In cells of the blood-brain barriers of rats, vitamin C worsened nitric oxide toxicity, while glutathione protected against it, similar to the original study that I mentioned at the beginning of this section. However, in this case, they used a nitric oxide donor that donates more nitric oxide in the presence of vitamin C, so that might have been the primary mechanism by which vitamin C was harmful. All right, that's kind of a lot to chew on, so how do we boil it down? The main common threads are these. Number 1, vitamin C is often, but not always helpful in cell-free systems using isolated components, especially if peroxynitrite is used directly or other oxidants are present to generate it, rendering that effect of vitamin C irrelevant. Number 2, In cells that are not brain cells, vitamin C may be neutral or helpful. Number 3, vitamin C is not helpful in any of the cells unless the concentration used is within the normal range of human plasma. And lastly, number 4, the last 2 studies and the original one that I mentioned at the beginning are the ones done in brain cells. In 2 of them, vitamin C was harmful and glutathione was protective. In the outlier, there was a source of cysteine to make glutathione. Overall, these results are consistent with the following principles as applied to studies of nitric oxide toxicity. Number 1, normal physiological concentrations of vitamin C are helpful, but supraphysiological concentrations can be harmful. Number 2, they are especially harmful to brain cells. Number 3, they are especially harmful when glutathione is not well balanced and when the concentrations of vitamin C might be the major source of oxidants. All right, and if all that was too much, here's the bottom line. Vitamin C appears to generate oxalate consistently, using 2,000 milligrams per day in apparently healthy people and using 1,000 milligrams per day in people who have recently been on a low vitamin C diet. In the latter, some people generate oxalate from 400 milligrams per day. All of these studies provided the vitamin C as 2 divided doses, and single doses would probably be even more likely to generate oxalate. The oxalate represents a failure in the 2-step process of vitamin C recycling. This implicates that glutathione are one of the factors in the suite of vitamin C recycling nutrients we're limiting. However, there are no studies that clarify what dose of vitamin C generates oxalate when all these factors are fully optimized. Oxalate generation represents the most sensitive stress in the vitamin C recycling system, and it is clear that high doses of vitamin C stress this system in people of average health in modern society. This may represent the point at which vitamin C could lead to a greater oxidation of the glutathione pool, but this is not well studied. The concentrations of vitamin C that kill cancer cells represents the point where vitamin C has clear potential to generate superoxide and hydrogen peroxide. These concentrations cannot be reached using oral dosing, but are routinely reached using intravenous vitamin C. In cancer patients, the pro-oxidant effects of high-dose vitamin C are necessary. These are supported by case studies, case series, and not randomized controlled trials. So there's no evidence-based medicine endorsement of high-dose vitamin C in the context of cancer. However, the general principles suggest that it may be of a special value in synergizing with pro-oxidant chemotherapy, but they also suggest that it could be valuable in any cancer that has vulnerability to oxidative stress. So that might have very broad applicability. Of course, I cannot endorse you getting any medical treatments, but this is something where you might want to talk to a complementary medicine specialist who uses high-dose vitamin C in this context if this applies to you. In these cases, you need to separate the glutathione and use it at the maximal distance from the IV vitamin C in order to support the host without antagonizing the anti-tumor effects of the vitamin C. Now, in sepsis patients, high-dose IV vitamin C does not generate high concentrations because it's rapidly consumed. And in dialysis patients, low doses are used and concentrations improve but don't exceed the normal range. High-dose IV vitamin C has been shown not to cause oxidative damage in healthy people, but it has not been studied whether it causes oxidative stress as measured by oxidation of the glutathione pool or impaired glucose tolerance. And arguably the most sensitive rise of oxidative stress in response to vitamin C is oxalate, which has been shown to increase because the oxalate represents a deficiency in the recycling, which is highly tied to the antioxidant network and the oxidative recycling network. Therefore, 1, those using 1,000 milligrams per day or more of oral vitamin C should pay closer attention to their glutathione hygiene in the suite of vitamin C recycling nutrients. Number 2, those using 400 to 1,000 milligrams per day should also consider this, especially if they have oxalated-related problems. Number 3, those using 2,000 milligrams per day or less probably do not need to worry about this, but some do. You would know by symptoms. Rarely, some people report negative symptoms in response to 200 milligrams per day. Number 4, practitioners using IV vitamin C for cancer should separate it from IV glutathione to reap the pro-oxidant benefits of the vitamin C. Number 5, practitioners using IV vitamin C in people not suffering from sepsis and at doses higher than 250 milligrams per infusion or 1 infusion per week should pay very close attention to glucose tolerance and measure the oxidation status of the glutathione pool in plasma, and should pay close attention to managing glutathione hygiene in the suite of vitamin C recycling nutrients and to oxalate. Okay, so on that point of managing glutathione hygiene in the recycling suite, these are some helpful tips. Number 1, make sure to consume at least 1 gram of protein per kilogram of body weight or at least a half a gram of protein per pound of body weight. Number 2, consider supplementing with 20 to 40 grams of whey protein with anywhere from 500 milligrams each to 10 grams each of N-acetylcysteine and glycine, or anywhere from 500 milligrams to 5 grams of glutathione. My personal preference is for whey protein supported by additional glutathione. I would stay towards 1500 milligrams of NAC or glutathione without a compelling reason to use higher doses, especially considered what I've done in other episodes on the potential for NAC to hurt the mucus barrier of the gut lining. But consider what I've put here as the top of the range to experiment with. Number 3, keep diabetes well treated and reverse type 2 diabetes if possible, and reverse any signs of emerging prediabetes. Number 4, keep any thyroid, adrenal, or metabolic disorder well treated. Number 5, eat a diet that keeps postprandial blood sugar under 140 milligrams per deciliter, which is 7.7 millimoles per liter. Within that constraint, eat freely of whole fruit and unrefined sweeteners, but use moderation and steer very far away from 42% of calories coming from simple sugar. Within these constraints, any increase in carbohydrate is likely to improve glutathione status by increasing insulin. Second and last, don't start a new milk habit leading into a new use of high-dose vitamin C. If you use dairy products habitually, however, no need to stop. And lastly, be mindful that NADPH status will be lower in individuals with glucose-6-phosphate dehydrogenase deficiency and in premenopausal women during the follicular phase of their menstrual cycle. But then also keep in mind that collectively, having some defect in energy metabolism at the level of carrier status, meaning a 50% bottleneck in a metabolic step, the prevalence of the 1,451 problems is approximating asymptotically at least 100%. Now, the most important nutrients in the vitamin C recycling suite are riboflavin, niacin, selenium, calcium, magnesium, glucose, and energy. The reliance on energy metabolism additionally implicates all the other B vitamins, all of the electrolytes, copper, iron, sulfur, and all the 1,451 metabolic problems that could hurt energy status. And it's important to have enough selenium, copper, and iron, but not too much. Excess selenium causes oxidative stress generally, and iron and copper in their free form both increase the autoxidation of vitamin C. My Vitamins and Minerals 101 Cliff Notes are a great place to start managing these nutrients. I have a comprehensive system for managing nutritional status focused on lab work outlined in the cheat sheet, and MasterPass members have access to these for free and can buy and others can buy the Cliff Notes and cheat sheet on my Substack. I'll put the link in the description. And then remember that this video is only available for free for 48 hours. After that, it is only available as a preview except to MasterPass members. So consider the MasterPass because you not only get access to this, you also get all the other premium content and you get all the ebooks for free. See the link in the description for how to access the MasterPass as well as the written and referenced version of this article and the Evergreen podcast and video.